Thank you very much. Ladies and gentlemen, it is such a pleasure for me to be here and to share with you the work that we are doing, really harnessing the power of genomics to enhance tree resistance. So climate change may pose two scenarios. The first scenario is that there'll be a negative effect on pathogens and pests. This means that we will have, shorter breeding cycles, maybe less insects. Maybe we're going to have unfavorable conditions and we're not going to have enough pathogens around, which would be great. That means that we would have increased productivity in our plantations. It means that we would have better wood products and, of course, economic gain. The other scenario is that we can have a compounding problem. We would have biotic stress, which is pathogens, insects, and we would have these abiotic stresses, such as drought, heat stress, cold stress. And this is going to render our trees even more susceptible. This is going to lead to damage of our trees, and then we're going to have a loss of ecosystem services. Can you imagine not having a picnic under a beautiful tree? And we'd have loss in terms of wood quality and wood products. And this also leads to a loss of carbon drawdown, which we really need our forests for. So if you look at records over the past 30 years, massive tree mortality is increasing. And the reports indicate that it is both biotic and abiotic stresses that are the cause. So what we really need are resilient trees. Resilience is the capacity to recover from such difficulties. Now, in South Africa, where I'm from, we rely on both eucalyptus and pine forestry. Eucalyptus is native to Australia and the surrounding islands, and it makes up 50% of the plantations in South Africa. In most cases, it is hybridized with Eucalyptus grandis. The pine species that we are using are actually native to Mexico and Central America, and we are using hybrids for improved performance. So, for example, hybridization for frost tolerance. So, what my team and I are really studying is how a tree is talking to a pathogen and how the pathogen is talking back. So, to really understand this molecular dialogue, we use transcriptomics. Transcriptomics, as was described very nicely by Steve, is that we are looking at the message that is sent. So this message is then translated into the chemicals that a tree produces, and this is a branch of study called metabolomics. Now really, this is all spearheaded by the production of the poplar genome, and thank you and congratulations, Gerald Tuskin. Soon after that, it inspired the genome sequencing of Eucalyptus grandis. And so we had a high-quality genome from which to derive much more information. So today, I'm going to tell you about a curious canker that affects our eucalyptus trees, a brutal blight that affects young eucalyptus leaves, and then a nursery wilt disease that affects pine production. So this curious canker is caused by a fungus that is known as Chrysoporte austrofricana. And I share this with you because it is a relative of the pathogen that causes chestnut blight. This has led to the fall of the American chestnut. So in this picture, you can see some of the trees have cankers. These cankers render the trees quite susceptible to wind breakage. We have these controlled inoculation systems. So back in our laboratories, we've got these kinds of experiments that go on. We can apply the fungus in a controlled way, and then we can look for those that are resistant and those trees that are susceptible, and we can do some comparisons. So plants make hormones. And what we found is that in the resistant tree, there was a higher level of this hormone, salicylic acid. So you would know salicylic acid is the main ingredient in aspirin, and it's almost like the resistant tree knew that a headache was coming, which was the fungus. It also had lower levels of jasmonic acid. So we also looked into putting this as a model of the dialogue. And what we identified was a difference in this gibberellic acid signaling. A particular gene is being downregulated in the resistant host at 3 days, while in the susceptible one, this gene is being upregulated. And so we tried to figure out what is happening and what is the fungus doing in the tree. The fungus itself is actually making this gibberellic acid, and it is promoting susceptibility. The resistant tree is able to stop this in its tracks. So we are first working on individual clones. And so we started moving into these structured populations. And here we had a family of a hybrid that was then replicated for us. So we worked with 2 industries back home in South Africa, and they've produced for us these replications that allow us to have about 12,000 trees. What this means is that we can go in and do in-field inoculations, and after a couple of months, we go back, and this is a type of cankers that form, and we measure the lesions that are produced. So we've been working towards this type of systems genetics approach, just looking first at our nursery type inoculations, our field inoculations, gathering all of the phenotypes, and then associating those phenotypic differences with the genetic differences. And the way we're doing this is we're using the 72,000 eucalyptus SNP chip that Dario mentioned earlier. But not only are we looking at the genetic level, we're also looking at particular metabolites that may be produced. And we're working with a colleague, Professor Almond Hammerbacher, to look at certain chemicals that may differentiate resistant and susceptible trees. Because, of course, it's very difficult to go in and work with 12,000 trees and go in and do those inoculations. Next, I'll tell you about this brutal blight. It's caused by a pathogen known as Teratosphera destructans, and in this picture, you can see the devastation it causes. The clone that is shown in the front of the picture is highly susceptible, and there in the background, you have a more resistant clone. This pathogen affects those trees that are grown in tropical and subtropical areas, and also has become a problem in the nursery. This is the distribution of the pathogen, and when it was identified in South Africa in 2015, the first thing my colleagues and I had to do was to really put together a protocol to do these repeat inoculations to mimic the kind of conditions to get the best symptoms. In that way, we developed a scoring system, so all the way from asymptomatic to leaf deformation. This way, we could then screen plants and know whether they are highly resistant up to highly susceptible. So again, we looked at the transcriptome and how these plants were responding. The resistant plants very early on seemed to recognize the fungus, activating the correct type of defense responses. Interestingly, the susceptible hosts were only activating these kinds of recognition signals a lot later. So we were very curious about this early line of defense, and what we recognized was that on the leaf surface, where you had the fungus growing, around those areas, you also had leaf wax that was degrading. So we looked a little bit closer at the leaf wax composition, and in one of the clones, we found that after inoculation with the fungus, there was a particular compound called cycloartenol that was increasing. And so we applied the cycloartenol, and we applied this first in a Petri dish, and we saw that it has an effect on pathogen germination, where it is suppressing pathogen germination. We also applied this compound onto the leaf surface, and we found that increasing the amount of the compound decreased the pathogen germination. to the point that if we treated a plant that was considered initially highly susceptible with cycloartenol, we then would find that it became more resistant. So we were able to change its particular ranking. I'll move on to talk to you about this nursery wilt, and it's caused by a pathogen known as Fusarium cincinnatum. In the nursery, it halts production of susceptible species to almost 50%. And in mature trees, you can find this kind of symptom where you have this pitch that is oozing from the tree. And out on the landscape, if you see this kind of red flagging on a pine, it is typical of the symptom caused by this pathogen. It is still prevalent in many countries around the world. And our big challenge was to identify what underlies resistance against this pathogen. And so we again had a controlled inoculation system where we are able to clip the apical bud or the tops of the little pine trees, and we could apply the fungus, and we could do comparisons with treated with the fungus and not treated with the fungus. And we had the experiment where we had RNA extractions done at 3 days, 7 days, and we looked at both a resistant and a susceptible species. So Pinus technomoniae is resistant to this fungus, and Pinus patula is highly susceptible. In the model, looking at the resistant interaction, what we saw that was really interesting is that at 3 days and 7 days, there is a type of consistent response. If you look at our susceptible interaction, very early on, you see that there's very little types of responses going on. But there's also, at 7 days, a terpenoid biosynthesis, which is quite an important part of defense, is actually being suppressed in the susceptible interaction. But it was really looking at what the fungus may be doing that got us to understand how the resistant tree is winning. We looked at the pathway that relates to agostral biosynthesis. So the agostral biosynthesis is important for the fungal cell wall integrity. And what we saw was that the resistant tree is actually inhibiting the fungal lipid biosynthesis. So from within the tree, there's suppression and compromise of the pathogen. And that is how we think that the resistant tree is winning. So we have been working towards this population where we are now examining using a precision phenotyping, where we are looking specifically at fungal biomass. So if we can count how much of fungus is present, we'll be able to get a better sense of resistance or susceptibility. We've also been working with field inoculations, and we also have a pine snip chip. So we have a tropical pine snip chip, and with that, we are now relating these phenotypes that we are collecting to the genetics. And so we have regions on the genome that we can hone in on that will be associated with resistance. So I've told you how we've been looking at this molecular dialogue, but the big question is, how do we begin to integrate these layers of information? And over the years, my team and I have studied various interactions with eucalyptus and different pests and pathogens. And what we're looking to do is now identify these so-called hub genes, that is central points that are being affected by different pests and pathogens. So we are layering information thanks to genome sequencing, the transcriptomics, proteomics, metabolomics, and even looking into the microbiome. And what we came up with is that central to all of these different interactions is that there's nitrate-associated genes. And this is going to be the subject of the next phase of our work, looking at functional genetic testing of selected candidates, doing some of the kind of work together with CJ to look at poplar knockouts and overexpression, and we can do some work in eucalyptus as well. So ladies and gentlemen, I've told you a little bit about the biology of resistance. It's quite complex, but we do look forward to using the outputs for DNA markers for resistance. And as Dario pointed out as well, genomic selection models will help us to do predictions for disease resistance. And we hope to achieve a tool for yield protection so that we have sustainable forests. I want to thank my lab back home. These are the people that make the work happen. And thank you to the funders that enable us to do the work. Thank you for your attention.